With that said, I would like to introduce our speaker for today. We have Dr. Vaughn Mancha connecting to us from not so sunny Alabama, but warm. He's here to talk to us about the field of epigenetics and a major disruption it is causing in the field of evolution. A recent news article reports current estimates are that approximately 1/3 of professional academic biologists who do not believe in intelligent design find Darwin's theory inadequate to describe all the complexity in biology. In this presentation, Dr. Mancha will describe how epigenetics best explains the biological complexities of genetic change and why it's so disruptive to Darwin's theory of evolution. Dr. Mancha has an MD from the University of Alabama at Birmingham. He has an undergraduate degree in biology from Florida State University. He's a board-certified family practitioner. He currently resides and is director of Ratio Christi in Alabama. He's also the president of the Daphne, Alabama chapter of Reasons to Believe. And with that, I will introduce Dr. Mancha.
Okay, well, thank you very much for that introduction. And so we're going to talk a little bit about epigenetics and what it is and why it's so threatening, basically, to neo-Darwinism. This first slide, as you see, is a little animated slide. It's a GIF, and it's actually a little bit summary of epigenetics, because what you see is that the DNA is wrapped up in these donut-looking shaped things called histones. And these little tags, which are epigenetic tags, will come off of those histone tails so that it can unravel. It's an interesting fact that if you took your DNA of every cell in your body, which is 40 trillion, and you have 6 feet of DNA in each cell, and you laid it end to end, it would go to the sun and back 300 times. That's 93 million miles. There and back, actually, it's 180 million miles. That means that our DNA how our DNA works is entirely controlled by epigenetic mechanisms and how it tightens up the DNA. We just used to think of DNA as a strand and everything had access to it, and that's absolutely incorrect. Epigenetics is the study of how behaviors and environment can cause change and affect the way your genes work. But unlike genetic changes, epigenetic changes are reversible and do not change your DNA sequence. That's the big difference. With Darwin, you mutate the DNA to make a new thing. With epigenetics, tags are put on the DNA that can come off and go back on, that causes a different expression without mutations. That is at the heart of the problem between neo-Darwinism and epigenetics. This is an article that I read 6 years ago, and I couldn't believe it because, I haven't gone through biology in the 70s at the height of evolution, and I thought that it was just a fact that couldn't be denied. And then Nature Journal, which is the most important journal out there. I mean, when people read a journal, 50% of the time it's going to be Nature. That's how much of a leader this journal is. And they came up with this title, Does Evolutionary Theory Need a Rethink? And I almost fell off my chair that they would actually talk about this. And it basically was an article discussing that the older scientists who are just strictly neo-Darwinists do not accept this new epigenetic information that's coming out because it threatens their old paradigm. In fact, at the end of the article, it said the mere mention of epigenetics evokes an emotional, even hostile reaction amongst evolutionary biologists. Isn't that incredible? They find this new stuff, you'd think they'd be happy about it, but they're hostile because it affects their religious viewpoint. Too often, vital discussions descend into acrimony with accusations or muddled misrepresentation this is very telling. Perhaps haunted by the specter of intelligent design, evolutionary biologists wish to show a united front to those hostile to science. So if you're not with neo-Darwinism, you're hostile to science and you're thrown in the bucket of intelligent design, which this is not even an intelligent design issue. So this is a very significant article to come out, and it's only been getting worse. At the introduction, this article was read as well. It's from the Federalist. And it said the field of epigenetics is causing a major disruption in the field of evolution. A recent news article reports that current estimates are approximately 1/3 of professional academic biologists who do not believe in intelligent design find Darwin's theories inadequate to describe all the complexity in biology. Now, you cannot find a Pew Research or anyone else that will tell you exactly how many people believe in neo-Darwinism and how many don't. But I will tell you, there was an article that just came out in The Guardian, and the gentleman that was for the newer evolutionary theory, including epigenetics, said, we're going to win this battle, and half the room broke out cheering. And the people that are cheering are young people. In other words, the young people do not have so much vested interest in neo-Darwinism. And they want to go where the money and the research is. So when you look at any of these college campuses and you look at the age of the people that are in the department studying epigenetics, there's not anyone over 30 years of age. To put this in perspective, there's only been 170 journal articles that even touch upon the concept of neo-Darwinism in the last 10 years in PubMed. But there's 120,000 journal articles about epigenetics. It's, epigenetics is steamrolling through, and neo-Darwinism has just been cast aside. Nobody wants to admit it, but that really is the case. Let's go back to the beginning. This is the beginning, natural selection, and how it works, basically. And this is the Origin of Species, first edition. You can get those for free, by the way. You just download them off Google Play. He says, if the conditions of life and the form undergoes modification, well, Darwin had no idea what the form is, and that's genetics. But he also said it had uniformity of character. So whatever this stuff that was causing change was uniform, and then it can be handed off to offspring by natural selection, preserving similar favorable variations. What's interesting is epigenetics can work with natural selection, but it's also working apart from natural selection because it doesn't change the DNA. The sequence, that is. I mean, it changes and it has tags on it, but the sequence doesn't change. Now, this gentleman here was 50 years before Darwin. His name is John Baptiste Lamarck. He coined the term biology, and he believed that our experiences and our environment could influence our germ cells and our inheritance. And he was resoundingly criticized for this, and neo-Darwinism specifically rejected this for 150 years. But it turns out he's right, because his very explanation of things, when you read about it, sounds exactly like epigenetics. Here's a gentleman that really wanted to put a nail in Lamarck's head, and this is Agos Weisman. He was an embryologist. He was a brilliant embryologist, but he did this stupid experiment where he took a bunch of mice and kept cutting their tails off each generation, saying that, well, if the environment is changing things, surely the next generation of mice should have no tails. The problem is he completely missed the interpretation of the experiment. He should have been measuring the amount of post-traumatic stress syndrome those mice were having for their parents having their tails off. And if he had measured that, then he would have gotten some results. But the way he made his experiment was oversimplified and just simply mutilation. Nevertheless, he called this the Weisman barrier. And as you see here on the far left, you have the germ cells like the eggs, etc. And then it spawns off the soma, like the organs, the liver, and everything else. But it always, he claimed that it always goes from germ cell to germ cell and never back from the soma to the germ cell. That doctrine is being held today by neo-Darwinists, even though epigenetics goes around this. It's uncanny, really, when you think about it. They would hold a theory of a 18th century embryologist as though that should be affecting modern genetics today. I mean, they get it, their oversimplification of the way things work has caused them to hold religiously to these concepts. Further adding to the concept of neo-Darwinism, which is also called the modern synthesis, is the concept of the central dogma made by the co-discoverer of the DNA molecule, Francis Crick. The central dogma, I mean, I believed it until about 5 years ago when I realized that epigenetics was going around it. I thought it was a bulletproof argument, but basically it says DNA goes to RNA, goes to protein, which in a large majority of the times it does, but there's also a large amount of times it doesn't. And I'll explain it a little bit. In fact, really, the majority of the time it doesn't, because the junk DNA does not fit into this model, and yet it makes up 98% of the DNA. So the central dogma really theoretically only works on 2% of the DNA. So with regard to neo-Darwinism, you look at the top rung of the DNA polymerase, and it's the thing that replicates the DNA, and it occasionally allows certain mutations to occur. And they say, well, you know what, those mutations are random, but that's the feed for neo-Darwinism, that these mutations add up. They're just happy mutations that just happen to be in the right sequence. We are learning, however, that polymerase is controlled by epigenetic repair mechanisms, and it's not random. It's a cellularly fixed mechanism with teleology to it. And this is all coming on, so that the concept of random genetic mutations are going out the door. They're being relabeled biased mutations. In other words, they're fixed in a particular area. I'll in fact discuss that a little bit later. So what happened then is one of the great fathers of evolution, who's John Maynard Smith in the early 90s, he took the Weisman Baer principle. You see on the top, the G goes to G, goes to G, goes to S, and they added it to the central dogma on DNA, and then DNA. You know, these are actually such a conflation of two ideas that are completely leading, but it misled a bunch of people for years. These concepts are absolutely not connected, and yet because he can make these stick figures look similar, he tied these two ideas together. What a disaster! But they were considered bulletproof for the longest time. So are mutations actually random? Well, the idea by neo-Darwinism is that a random mutation happens, and then a bigger muscle or bigger fin is made, and then you can see the penguin's got a bigger fin, so he goes up there and he gets the pretty penguin, and they have kids together because of his mutations. And then there's a lot of arm swinging, etc. You know, a lot of, oh, well, just give it a few billion years and this will happen. The just so stories. So that's what Roger Kipling talked about, just so stories for little children, where, you know, you tell them how the leopards got their spots. And Stephen Gould of the Punctuate Equilibrium fame said that, actually tagged the Darwinists with this tag. He said they're just looking for just so stories where a micromutation might have occurred, and then with a lot of arm swinging and billions of years, it might turn into something. And on the far right here, we see a just so story being applied where the bear has picked up some tools and learned how to take a fence apart because which is causing natural selection to a higher IQ. Now that seems silly, doesn't it? But I'm going to present a paper in a little bit that's just as silly as this. Okay, so this is with the Central Dogma. I think you recall that slide back there. You had DNA, RNA to protein, and nothing else, right? With epigenetics, you have the DNA, and it's wrapped up in those donut-like things. You remember that very first slide, how the donuts would open and shrink, and it's got these tails on it that you see, the green, yellow, and red that affect how these things are wrapped up. And this is DNA to the sun and back 600 times, an incredible amount of compaction that's going on. And that determines how the DNA is read. Again, we see here the compaction of the DNA that's going on because of epigenetic mechanisms. The DNA is not just some string that's sitting out there being subject to the central dogma. I'm going to show a little introduction to epigenetics so you can get a better visual. Can y'all hear? Can y'all hear this okay? Hello, can anyone hear me?
Yeah, we're hearing just fine and we're seeing the slides.
Yeah, okay. Well, no, but you're seeing this this presentation.
Yes, we're seeing it just
Okay.
fine.
Okay, thank you.
But we're not hearing any sound with it.
Okay, there is a button that you have to hit under the sharing slides aspect.
Okay, I have been hearing it just fine all along.
And how about everybody else?
Not on Zoom, but we're not.
Well, I can. Okay, I just hit a button that should change things.
Because our DNA is very long, cells package up their DNA together with protein.
Are y'all hearing that now? Can somebody tell me if they can hear it?
It was with a lot of echo.
Let me try one more time.
Proteins known as histones. The DNA and protein complex is
Yes, we hear it now.
DNA is wrapped around histone proteins, forming repeated units of nucleosomes that look like beads on a string. Chromatin is condensed further to form a chromosome. We have 46 chromosomes in total, stored in the nucleus. The core of one nucleosome is of DNA wrapped around a histone octamer that consists of two copies of the major types of histones, H2A, H2B, H3, and H4. This organized DNA protein complex allows the cells to regulate what genes are expressed and when. All the cells in our body contain the same DNA sequence. Skin cells, muscle cells, and liver cells contain the same DNA sequence. Yet all these cells have different structures and functions. The reason is that different cells only use or express certain genes. Within each cell, the DNA and also the histones can be tagged by tiny chemicals that modify gene expression. These chemical tags cause some genes to be turned on and some genes turned off. For example, muscle cell genes and will turn off genes that a liver cell requires. Similarly, the liver cell will have genes for muscles turned off and genes the liver requires turned on. These modifications are known as epigenetic modifications. Epi is Greek for above the genes, so modifications above the genes or on the genes. Epigenetic modification brings about lasting changes in gene expression. Let's have a closer look. Here is a normal human cell, and we are pulling out its genetic material. The DNA is wrapped around nucleosomes, which are made up of histone proteins. Coming off the histone proteins, we have histone tails. DNA is made up of a combination of 4 nucleotides: G for guanine, C for cytosine, A for adenine, and T for thymine. There are 2 main epigenetic modifications. The first one we will talk about is DNA methylation, where a methyl group is added directly to a cytosine residue that exists in a cytosine guanine sequence, or CpG sequence for short. So for example, here we have many CpG sites that make up a CpG island. And here the cytosine are methylated. The methylation of CpG sites in promoter regions is associated with gene silencing. The second type of epigenetic modification is histone modifications. This is where acetyl or methyl groups are directly added to histone. This will modify. For example, histone acetylation correlates with transcription activation, and histone 3K27 trimethylation with transcription repression. Genome-wide patterns of DNA and histone modifications, or epigenome, are established during early development and are maintained during cell division. In cancer, these patterns are altered and disrupted.
Okay, did that seem to make sense to folks? Okay, so this is just kind of a reiteration of what you just talked about. You can see the histone yo-yo looking blue structures, and the DNA wrapping around it. And when the DNA is tagged with methyl groups, little red groups, if it's not tagged, then the DNA strands are more open to being transcribed. So you can imagine if the stretch of DNA wants to make more cells that for your liver, it will open up these stretches and change the amount of methyl groups. But if it doesn't, but say your liver's finished being constructed as a child, it will slow down by tightening up and putting methyl groups on it. On the very bottom, you can see it, so it's tightened up. This completely changes our concept of the way the DNA works. It's not a mutation and a change in things, but it's the same DNA doing this different things for different organs. This is one of the big problems with neo-Darwinism. And one of the reasons there's a whole branch of study called Evo-Devo that separate from evolution, because they were interested in the fact that you had 200 tissue types in your body, and yet each cell had the same DNA. How could Darwinism explain that? It couldn't. Epigenetics fully explains it. So the whole field of Evo-Devo is that we are into epigenetics, all things epigenetic, and we don't care about neo-Darwinism. And this is sending a shudder down the spine of all these neo-Darwinists. So you have the body in the middle called the phenotype. We call it genotype as such your DNA, and then the phenotype. This is a terrible way of looking at things, but this is one of the ways that neo-Darwinism forced us to look at things for years. Their phenotype is not necessarily made by the genetics on the right. Genetics and neo-Darwinism is made by DNA sequence variations, that the letters of the DNA is changed. And that's what causes a new phenotype. However, according to epigenetics, the environment causes the changes. That's what John Baptiste Lamarck originally started with. And oh, by the way, Darwin really did too. He said of the two things, natural selection or the environment, he felt like the environment was more important, but nobody really paid attention, and they ignored his the parts of Lamarck which he really liked. And so that really set neo-Darwinism down a path of hardened genetic interpretation, which is why it's failing. So according to epigenetics, it's epigenetic modifications that causes these things, and it changes how the genes are turned on and how they're turned off to change the phenotype. So it's not mutations, rather how you play the DNA that counts. So this is a rather interesting article. It came out 8 years ago, and it's Nature Journal. You know, they're considered the Bible of evolution and everything. And you can imagine the kickback that happened when they came up with this title. It said Epigenetics, the sins of the father. Well, that's the sins of the father is right out of Exodus 20. And God says, if you hate me, I will visit the sins of the father to the third and the fourth generation. So basically, our behavior can cause behavior to be sent down generations. And what was happening when this article came out is they were discovering that things like rat experiments where they stimulated the electricity on a paw and gave it the smell of a citrusy smell. After 3 generations down, they could give it the citrus smell and the rat would go running. So in other words, behavior is being sent 3 to 4 generations. And they were discovering this in all different animal models. So somebody said, you know, that sounds like we're out of the Bible. And so they put this title up. And of course, the evolutionists really loved it. So this is that verse in Exodus 20 that if the sins of the father go to the third and the fourth generation. And what this does is this makes the 10 Commandments are not just a behavioral guide for you but it's really more so that you won't injure your offspring epigenetically to the third and fourth generation. So it's not just a series of good things you ought to consider, but it's a way to help your genes do better. But again, it's controversial because it harks back to the discredited theories of John Baptiste Lamarck. In fact, this is what the guy said in Sins of the Father. He pointed to John Baptiste Lamarck, and he proposed originally that acquired traits can get passed to the future. But this makes so much sense in so many different ways. Like a bird, for instance, giving its song to its baby's birth. That's not a mental thing, and the birds are born that way, chirping that way. These things are actually ingrained on top of their DNA because there is no DNA sequence that makes a bird have a particular song, but there's epigenetic tags that can promote those particular songs. Going on in this article, too many modern biologists say it's scary sounding. Those who work suggest that such inheritance does indeed happen in animals. If it's true, he says, why hasn't this been obvious to the brilliant researchers in the past 100 years of genetics? Isn't that amazing? Because people are so against it, they couldn't see it. And it was right in front of their faces. This is just an illustration of how epigenetic inheritance of acquired traits through DNA methylation, little methyl tags are put on it. And you can see the trigger on the bottom left, and then it puts a little methyl tag on the DNA. And there's other things working together with it. This is actually kind of the picture of that rat study I was telling you about. There was the odors, the hormones, and the cytokines and other things that went into getting the rat to have a reaction. So basically, the environment can affect things that go beyond the grave. And this is another rat study that they did. This was actually based upon a Danish study they did of these ladies that had been under the Nazi occupation. They discovered that the ladies that were during the occupation were in starvation conditions and delivered their babies, that those babies were underweight, more schizophrenic, and more health conditions. Whereas if the baby came after the occupation, it was healthier. And so this showed that the environment can change the inheritance of things. And this shows it here where there's a certain type of mouse that that because of, it's called a goosey mouse, but because of particular epigenetic trends it received, and when it ate before delivering, its mice would be fat. Now this is a amazing study that came out. Darwin rode on the boat, the Beagle, to the Galapagos Islands so famously, and he started his theory based on the finches, actually. By the way, I was just looking on Facebook. You can actually go to the Galapagos for almost nothing right now. Of course, it's not just the Galapagos, so you got to go over there and support Darwin. Anyway, he came up with this. This is how he came up with his concept of natural selection, that the beaks of different finches on different islands were changed. And he didn't know why that was. But he just thought that once they were changed, they passed it on, and that was probably because they were more fit. So this doctor Skinner decided to study this with regard to epigenetics. And he wanted to compare the epigenetic changes among several closely related species of finches, which is a well-known example of adaptive radiation. That's basically speciation. And he showed that the epigenetic changes contributed to the molecular basis of the evolution of these finches. That's a huge statement right there. I mean, they were saying for 120 years, none of this stuff went forward. And yet suddenly we discover that epigenetics has contributed to these changes. And the title of his article is Epigenetics and the Evolution of Darwin's Finch. So there was, what's interesting, that there were relatively more epi mutations than genetic mutations. So epi mutations are tags in the different types of epigenetic tags. And he was seeing a lot more of these tags than he was seeing genetic mutations. I mean, there's genetic mutations in everybody, but he was really seeing more epi-mutations, just indicating that it's epigenetic changes, not mutations, that's causing the change. So it suggested that epi-mutations are a major component of the genome variation during evolutionary change. And it goes on. However, genetic changes alone are limited in their ability to explain the phenomena, ranging from the molecular basis of disease to aspects. So basically, as we do these studies on epigenetics, we're reversing our thoughts on Darwin, because it's not genetic changes through mutations that's causing things, it's epigenetic changes. So there's Darwin's quite upset, and Lamarck is on the right. He says, I'm coming back. And he should. He was maligned for 200 years, and he was in fact right. So this is kind of a review of what we were talking about. A in the top left, you have little red tags put on. That's the cytosine. They're epigenetic tags. You go to the top right, you have the histone things, which are looks like little donuts with tails. And those tails are subject to modification, which can change quite a bit. And they're very specific. Look at that one modification that's got a purple triangular flag on it. They know in a study of a particular rare brain cancer, which is horrible for kids at a very young age, that one of those histone modifications is off, just one. So one change, if it's done wrong, can do quite a bit of damage. So moving down to left bottom is what's called long non-coding RNA. This is the junk DNA, which, by the way, neo-Darwinists ignored for years and years. Yeah, 98% they thought of our DNA was junk DNA, and they would chuckle, God made it just to confuse the creationists. And suddenly their foot is so far down their stomach, it's coming out their behinds. So this long non-coding RNA is causing epigenetic changes as well in how it interacts. I'll give a little show about junk DNA.
In 2001, researchers of the Human Genome Project determined that only 2% of human DNA are genes, the sequences that directly code for the proteins our cells use to carry out their functions. The remaining 98% of the genome was concluded to be a biological wasteland. But it turns out that this supposed junk DNA is crucial for controlling gene activity, often acting as switches to turn genes on and off. These switches determine where and when a gene turns on. When a regulatory molecule binds with its corresponding switch on the DNA, it will cause the switch to flip and the gene to be activated. With the regulatory molecule bound to the switch, the gene will be expressed in greater amounts. When the regulatory molecule detaches from its switch, gene expression will stop. Typically, the gene will be surrounded by many switches that allow the gene to be turned on and off under the control of different regulatory molecules.
Okay, so this was just a brief explanation of junk DNA. This is in large part why why Dawkins wrote his book, The Selfish DNA. He could not explain all that DNA that didn't seem to have function. And he just said, well, it's selfish. That's why it's there, which is a ridiculous reduction. And then he'd run around and he'd say, creationists think that God made 98% junk, and he'd laugh and laugh. Well, he's not saying anything these days. He's in fact hiding. What's fascinating is, okay, do you think the neo-Darwinists were happy to find out what junk DNA is? No. They actually fought it tooth and nail. They said, well, it's junk, but there's only about 11%. And then we kept doing experiments. There's not a day that comes out that a new use for a particular piece of junk DNA has come out. It's up over 65%. But the guys that originally discovered it, called the ENCODE project, said it was at least 85% was non-junk, which the neo-Darwinists absolutely lost their brains over. So the more and more we study it, the more and more we realize it is useful. This is a science journal which was discussed the ENCODE project, who wrote the eulogy for junk DNA, and they lost their minds. There are some really nasty journal articles that came out where they just, in fact, they would write that one article they wrote where they just slam-basted them, and then there was only 2 people that would put their name on the article. The rest of them put their name hidden in the article because they didn't want to let it be known that that was them. That shows you how sneaky and these guys were. Here's the guy that really disproved it, although he blew the study. This is Francis Collins, who did the Human Genome Project, who ignored junk DNA. And he finally said at a conference that he said it was he said in terms of junk DNA, we don't use that term anymore because I think it was pretty much a case of hubris to imagine we could dispense with any part of the genome. I mean, listen to that. It was hubris, and yet he messed with 11-year study, $7 billion, trying to support basically theistic evolution, only to fall flat on his face. And then he said it was hubris, and then he pulled it out of his first book, by the way, which was supposed to be about God and DNA. And he ran down creationists, but then he went back and pulled it out of his book instead of letting it lie. So he went on to say, as if we knew, he said, most of the genome we used to think was there for spacer stuff turns out to be doing things. So here's the view now. You've got the Central Dogma on the far left, which was inadequate, and now you go add the junk DNA, and it changes things. You see that there's non-coding spaces and introns, and those introns can take say the far intron on the far right, that little grayish space between the 2 red genes. That intron can change the way those 2 genes are spaced to make up to 30,000 genes from those 2 sections. So it's not just fixed genes, but we're talking about scrambled genes due to this junk DNA. So in fact, that's another thing that completely disproves neo-Darwinism. Because neo-Darwinism cannot tolerate a gene that does not make DNA according to the Central Dogma. The Central Dogma on the far left, DNA to RNA to protein, that's the only DNA that's working. The rest of it's junk. Therefore, the junk DNA being proved useful is probably the biggest fault or fail of neo-Darwinism. There's some more of the non-coding RNA that's coding the DNA. Now, the ones on the two left on the 2, those have been around for a while. We have small nuclear RNA, small interfering, microRNA, Kiwi interacting, all kinds of different acting RNAs that we're just now learning what they're for. So junk DNA is involved in cellular function, including cancer, but we ignored it, laughingly saying God made it to test creationists. And this is an example of junk DNA being shoveled around in the cells. So Lamarck's junk DNA helps us respond to environmental stress. As well, one of the interesting things is the fact that one form of junk DNA, which is considered a virus, is very ancient in certain DNA, which includes primates. And so but this is probably the strongest argument for common descent, and that is that primates, including humans, chimpanzees, and others, share a bunch of different virus DNA that they believe got stuck in the DNA. However, that argument depends upon that DNA being junk DNA. So, but they can't really use that argument, can they? Because that junk DNA also has a great deal of function. So the argument of design comes into it, because this DNA that was in these primates that in some cases go back 5 million years also have a design aspect to them. So this argument actually cuts both ways. If you want to say it from the evolutionist point of view, then you're going to have to realize the fact that you missed the mark by 98% by calling 98% of things junk DNA. So, in any event, this is an interface of the syncytial trophoblast and the chlorotrophoblast within the placenta. That blue structure in the middle, if you see it, has a bunch of nuclei in it and these big membranes that wrap around so that they separate the mom from the baby. And those little islands of white is where the blood is able to move nutritional aspects. It's an incredibly complex organ, and it arose partially because of certain viruses. So when you look at non-coding RNAs, which is the part that, the non-coding DNA goes to non-coding RNA, it's a very complex network of gene regulation. A large portion of humans' transcript is bidirectional, producing non-coding RNA. It can overlap with the transcript of protein coding regions, it's, in other words, it's got almost an infinite potential complexity. This going back to this picture here, you see, because of the way the non-coding sections can move around, it just creates an incredible amount of complexity. And in this article, How the Genome Got a Lifespan, what we wish to highlight here is the contrast between DNA that is mutated versus chromatin that is reshaped after environmental input. The mutation bears no functional relationship to environmental exposure, whereas the change in shape is called forth in some functional specific way. It's a logic of imprint, not one of error. This really says that it's a logic of imprint, tags put on there, not one of DNA errors. So this is coming out, 7 years ago? And this statement would have been, they would have shot him in the head in a back alley when I was in college. You couldn't make this sort of statement. So they're basically saying it's a difference between epigenetics and neo-Darwinism. Now, this is an interesting article. It's really not related to epigenetics, although it is. I got into a challenge, actually, I think it was an atheist on a biology group somewhere. And he was just swinging around the arm saying that genes come around in evolution. So I said, show me one. Show me a journal article proving a gene coming into existence. And so I started looking, and I mean, I'm talking about looking through 1 million different journal articles on PubMed. But fortunately, you can do searches and tie it down. But all I could find was this one journal article that actually tried to tackle it, and it was so wanton, it's really funny. In fact, the very last line in the article said, "One can imagine," and I put John Lennon, imagine, because that's what they wind up forcing you to want to do, is imagine. And the name of the article is Darwinian Alchemy, Human Genes Coming from Non-Coding DNA. That's the junk that everyone looked at and said, oh, it's junk. And they said, well, it's the only place we can find a new gene, so maybe it's coming from there. And then they make this statement that's unbelievable. How could all these pieces fall into place through the random process of mutation, recombination, and neutral drift, or at least enough of these pieces produce a protogene, that's a beginning gene, that was sufficiently useful for selection, Darwin's natural selection, to take hold? And this is one can imagine. Imagine alchemy. No, what he's saying is how in the world could have all shuffled together? It just really is very honest in this statement. How could these pieces just shuffle together? Because you have to have an initial protogene before natural selection can work on it. But this is making the proposition that it all occurred before natural selection, which is completely against neo-Darwinian natural selection. You got to have the fixed thing before you can cause natural selection to act on it. So he says one can imagine, and in fact he says, imagine a small protogene coming around and then getting more complex. That was their mechanism. After reading through the stupid article, the mechanism they gave is to imagine a small protogene and then it got more complex with no mechanisms whatsoever. Now, this is an interesting article that came out this year. Two really just home run hitting articles have come out that has really changed things quite a bit. And I'm going to discuss this first one. This was actually from Nature Journal in January. And it says random mutations are an important part of the theory of evolution by natural selection, in which mutations give rise to adaptations that are passed on to offspring and alter their chances of survival. This study challenges the evolutionary theory that DNA mutations are random. Well, that's incredible right there. They're challenging the evolutionary theory of basically 120 years. And this is a Nature journal. How did these guys get up there? Why didn't they shoot them? You know. And then he says, we always saw mutations as basically random across the genes. It turns out that these mutations are very non-random. It's non-random in a way that benefits the plant. He's talking about this plant they're studying. It's a totally new way of thinking about mutation. That's an understatement right there. In other words, mutations are not random. These findings add a surprising twist, i.e. destroys Charles Darwin's theory of evolution by natural selection. Natural selection cannot be natural selection unless there's random mutations. So if the mutations are mainly non-random, then there's not natural selection. They go on to say, since the first half of the century, evolutionary theory has been dominated. I can't say that anymore, but it was dominated when I was in college by the idea that mutations occur randomly with respect to their consequences. We conclude that epigenetic mutational bias, okay, this is the epigenes can actually bias the mutations. Now, earlier I said epigenetics doesn't change the DNA. There is this exception that it does cause certain mutations, which are very, very quick and not as widespread, but they do cause wide effect, and they're called epigenetic mutational bias. Challenges the prevailing diagram. That mutation is a directionless force in evolution. Again, let's say that again. Mutational biases challenges the prevailing paradigm that mutation is a directionless force in evolution. A directionless force in evolution. I'll say it again, a directionless force in evolution. That is neo-Darwinism. That's why people love it, because it's a directionless force. And you don't have to fess up to a directionless force. There is no God with a directionless force. We're not culpable with a directionless force, are we? That's why they love it. That's why they loved it since the 1800s. Our discovery yields a new account of the forces driving the patterns of natural variation. By the way, they're calling it natural variation, not natural selection. Challenging a long-standing paradigm regarding the randomness of mutation. I'll say that again. Challenging a long-standing paradigm regarding the randomness of mutation. That's just incredible. This, if you get a chance, this is the one. There's two articles, and I'll send them to you. If you email me at Vaughn Mancha, first initial, last name at Gmail, I'll send these two articles to you. These findings demonstrate that genes subject to stronger purifying purifying selection, i.e. natural selection, are maintained in epigenetic states that underlie a significant reduction in the mutation rate. In other words, natural selection is being taken over by epigenetics. You remember those pictures of the epigenetic tags that were put on those donut-looking things? Because it tightens them up, it reduces the mutation rate, which is not a part of Darwin's natural selection. That mutational bias is the primary force behind the patterns of sequence evolution around the genes in natural sessions is supported by analysis of allele frequencies. In other words, we're proving that it's epigenetics that's causing these bias mutations. And these bias mutations are, in fact, you just don't see the word random mutation found in PubMed anymore. Nobody uses that. Everyone realizes it's biased. And because it's a specific cellular mechanism that's causing it, it's teleological. And so you can't, the basis of neo-Darwinism is lost. In other words, the areas in the DNA we have been claiming were changed by random mutations followed by natural selection were actually being changed by epigenetics without natural selection. I mean, that's huge. So here's an example of epigenetics and how it can work without necessarily causing mutations, but just in the way it's set up within the DNA. And this is the making of the ant cast. On the top left, you see this graphic of Conrad Waddington's diagram. Conrad Waddington was the gentleman that discovered epigenetics back in the 1940s. At the same time, they had just released the modern synthesis, or the neo-Darwinian modern synthesis. And he believed that a ball kind of rolled down and caused different valleys to cause different shapes and phenotypes. And on the bottom, you can see a miner, a soldier, and queen. And all these different shapes, all these different phenotypes, if you will, have the same genes. So how can they have entirely different phenotypes? It's because of methylization, epigenetic, Lamarckian epigenetics. And the same applied to the finches that we looked at earlier. So here's kind of a GIF showing the epigenetic. There's your DNA and epigenetic tags. And as a result of the way it puts its tags on, you get different tissues. Same genes, different tissues due to the different epigenetic tags. Different outcomes, different phenotypes, same genes. This is an article written by Dennis Noble. He said, "What future for evolutionary biology? The illusions of the modern synthesis, the illusions of the theory of evolution," is what he's saying. So the extensive range and depth of the 20 commentaries on my article confirms that something deeply wrong has gone wrong in biology. I wrote all parts of the neo-Darwinian discourse encourage the use and acceptance of other parts. I repeated this in a target article because the expression of the difficulty of trying to break out from the attractive simplicity. Look at that. He calls it the attractive simplicity, and that is the problem with neo-Darwinism. It's so simple. This is not E equals MC squared. I mean, genetics is vastly more complex, especially with the overlay of epigenetics. And yet they want to teach the same thing that, mutation occurs and it's, natural selection works. I mean, these are really old terms that need to be dismissed. He's saying students not being taught about the new stuff that's coming along. In other words, they're being taught about the central dogma as an illusion. I was taught that, the central dogma. Of course, it hadn't been destroyed by epigenetics. It said replicator vehicle distinction has to be abandoned. Lamarck needs to be reinstated.
Do you want me to pack this? Do you want to do this now?
Hello?
I'll do it tomorrow. You know what?
He said, I must be one of the first to unravel that incredible story in a detailed historical way to ask how so many brilliant scientists could have been taken in by what took to be the established truth. Evolution as an outcome of a myriad of mechanisms and processes that go beyond reductionist one-way causal views and require a nested view of organization. Illusions like the Central Dogma have both impeded and facilitated scientific inquiry. It was a huge mistake for biologists to eschew teleology. Now, we threw teleology out the door. When did the modern synthesis go wrong? Historically, it did in two stages. First, it uncritically accepted Weisman's barrier dogma in order to exclude inheritance of acquired characteristics. That was done in 120 years ago. You know, and his experiment was just cutting off mutilating mice, which is not a critical experiment at all, and yet people just accepted it. Second, following Crick's formation of Central Dogma, and that was formulated in 1970. And DNA, RNA, protein is only partly right, but it's wrong 98% of the time as far as junk DNA. And the growing convention that the absence of the other it vindicated the Weisman barrier, and I showed you that graph where the two were put together. This is a book I've been reading by Peter Ward. He lost, by the way, debated Peter Ward and is good friends with him. He's very impressed with him. And if you've read his book about the rare planet, he quotes Peter Ward like crazy. Peter Ward wrote a book called The Rare Earth Hypothesis, and it's standard reading at college. And Ross is a big fan of that book. And you can download a free PDF. He's also he's a paleontologist, and he realizes, like Stephen Gould, that Darwinism does not explain things, that Lamarckism explains things, and so that's why he wrote a book called his book Lamarck's Revenge. He said today a revival of Lamarckism, and basically he's talking about his epigenetics, the previously mentioned neo-Markianism, is causing many biologists to admit there's a major revolution and evolutionary theory might be at hand. The now favored theory, Charles Darwin and Darwinians who followed him, that is the major process of evolution, is driven by natural selection combined with genetic change by mutation. Epigenetics posits a different set of circumstances can drive evolutionary change, and that both Darwinian as well as epigenetic-driven changes can proceed simultaneously. But the question is still unanswered, is simply knowing which predominates or when. I love this paragraph. But on occasion, even seemingly irrefutable scientific principles, i.e. the neo-Darwinism, and paradigms, again neo-Darwinism, do falter because of revolutionary new discoveries that cannot be explained by old theories. That's an incredible paragraph. I mean, it was irrefutable. I mean, it was a fact like gravity, you heard people saying. And now they fail because they can't explain epigenetics. If you look at PubMed for epigenetics, and I mentioned this earlier, if you put in the modern synthesis, which is the modern or neo-Darwinism, there's like 170 hits. That's only 170 hits in 10 years. You put in epigenetics, that says 88. There's really closer to 120,000 hits. This is irrefutable scientific proof that epigenetics is for real, but it's also proof of why the scientists are moving away from their old simplistic views of neo-Darwinism to try to accommodate epigenetics. And they're basically, they just have to give up the old stuff. And on the top right, Lamarck says it right. Old Darwinists never die, they just fade away.